It's my pleasure to welcome all you participants in this webinar on the oncNGS project that we Sensano lead within the Horizon 2020 framework of the European Commission. My name is Mark van den Bulke and I'm the head of the Cancer Center at Sensano. Before starting, I would kindly ask everyone to mute his microphone during the presentations. We foresee in total 4 presentations, one after the other, and we will take some questions at the end of the session. We have a help desk installed to assist you in case that there would be any problems with the connection, and we encourage you to use the chat to communicate your problems, and we will try to help you out. Today we are here together for the open market consultation of the oncNGS pre-commercial procurement. We want during this session to give you some key information on the oncNGS PCP and get already a first feedback from you. Myself, I will give you just a general overview of the project. Luca Mazzarella will highlight the importance for patients and the interest of clinicians. Pierre Sujobert will tackle some of the challenges. And finally, Rossana Alessandrello will explain in detail the oncNGS PCP process. We expect feedback from the market by questions either through the chat today, through the oncNGS contact facility on our websites, and we will also forward you a questionnaire in a couple of days it will be opened that will allow you to give your inputs on what is really the importance of oncNGS and what questions you have on the tendering. And we will summarize all these questions in a frequently asked question note that will be available to all of you on our website. As you know, cancer is a major public health challenge in Europe and worldwide, but in Europe with nearly 3 million new cases and 1.3 million deaths in 2020. Cancer incidence will increase due to the effect of our aging population. Every year, cancer changes the lives of patients and their relatives and affects society at large, with an economic impact of the disease estimated around €1 billion annually in Europe only. Earlier this year, the European Beating Cancer Plan and the Mission on Cancer were launched. Both represent a holistic strategy at the European level to reduce the cancer burden and stressing the importance the European Commission is giving to beat this disease. oncNGS is focusing on providing a solution for patients with an advanced cancer where standard practices are not possible anymore or have very little benefit. The treatment in such case often benefit from a more tailored, stratified approach, the so-called personalized or precision medicine approach. The final aim of such stratification is to more accurately define which patients benefit from a particular treatment and who is not. This may greatly reduce the burden of unwarranted treatments and favor the quality of life of patients. The challenge the oncNGS buyers want to address to you, the market, consists of providing an efficient molecular DNA-RNA profiling approach on tumor-derived material in liquid biopsies by means of a pan-cancer tumor marker NGS analysis integrated with an ICT decision support system and including test interpretation and reporting. We are 8 buyers from 5 partnering countries in this PCP, and we come from Belgium, France, Italy, Spain, and Germany. As an example of a particular application of the oncNGS solution, we would be able, having this solution at hand, to create a possibility for clinicians to somehow match their patients with other patients that demonstrate a similar molecular profile as resulted from the oncNGS analysis. Combined with other clinical info, the treating clinician could then get a better view on what treatment is best suited for his patient. Moreover, based on these elements, it can then be envisaged to enroll the patient in a particular clinical trial. oncNGS indeed should facilitate to install such trials at the European level, which will allow that outcomes of the precision medicine-based treatments can be better evaluated as increased statistical power would be gained by having larger trials. Some general information on the project. So the overall aim is to develop an integrated solution for testing, analyzing, reporting, and storage of next-generation sequencing medical data within routine healthcare diagnostics. The total foreseen budget is more than 12 million, of which 90% is coming from the European Commission contribution. 70% of the total budget goes to the PCP itself. You can find more info on the call at the Cordis website and on the project as a whole on our website. Now, who are we? Within the oncNGS consortium, the buyers, or also called the procurers, consist of 8 partners coming from 5 countries. From Belgium, we have Scienzano and the Institut Jules Bordet. From Italy, we have representatives of the Alliance Against Cancer. From Germany, we have Charité Hospital and the Ludwig Maximilian Institute as partners. From Spain, the Catalan Institute of Oncology, and from France, Institut Curie and the Hospice Clinique of Lyon. Scienzano is acting as the lead procurer. In addition to the buyers, we have in our consortium 6 institutions who will help us addressing all the issues that are important for conducting the PCP. We have Aquas, who has vast experience and expertise in the PCP process, IPSA and VAIO, who will share their scientific and clinical expertise in personalized medicine in cancer, l'Institut National de Cancer as the focal point on cancer in France. The Clerc and partners will support us with outlining all intellectual property and regulatory aspects. And finally, we have the Belgian Cancer Register to help us to address registration compliance and needs. In the next slides, I've just listed all the partners in the consortium. And in the following slides, I will just pass through rapidly to give you some background information on each of the buyers, what organization they represent, their role, their experience, and their interest in the oncNGS project. Just to give you a broader insight on the overall group of buyers. I will not go through all the details. You will have all the slides available at our websites, but all of them are important players in the field of oncology, both in their own country as on the international level. All have a major interest for the project and commitment to the outcome. So Scienzano ourselves, we are the National Institute of Public Health. We are the coordinator, and we are involved in the standardization and testing and reporting, guaranteeing high-quality NGS tests for all patients at the Belgium level. Then we have Jules Bordet, is an OECI-accredited comprehensive cancer center. It has a vast experience in research, and one of the key features of the Institut Jules Bordet is the close integration of research and medical practice. Then we have the Alliance Against Cancer. This is the largest Italian organization for cancer research. It hosts in total 28 institutes, and their primary aim is to promote the network among oncological institutes pursuing clinical and translational research. ACC is focusing on major cancer types, and it aims to, by genomic characterization of tumors, offering an enormous number of opportunities in clinical applications. Then we have the Institut Curie of France. The Institut Curie is a composition of multiple institutes. It has a hospital and research center. It's localized in Paris at 3 sites. And it's been a pioneer in the field of precision medicine with the first randomized trial worldwide. And the genetic molecular platform of the Institute has been granted by the INCA, one of our other partners, to perform genetic next generation sequencing for clinical use. Then we have the Hospice Civil de Lyon. They are the second largest university hospital. They make part of the second largest university hospital in France. They have 14 multidisciplinary or specialized establishments, and they provide a wide range of services. And the network providing expertise in all these disciplines, both medical and surgical, but also very important to them is that they also perform research, are very much interested in medical innovation, as well as disease prevention and health indication. Also, the HCL has a label from INCA for molecular diagnosis of tumors. Then ICO is the Catalan Institute of Oncology. The approach there of the disease is very comprehensive, combining all in one organization, prevention, care, specialized training, and research. ICO is built out of 5 centers, and the ICO is the leader in cancer care in Catalonia with a high international recognition. It has a very strong research program in very different fields, and the programs are fully equipped in order to meet the needs of the investigators. Charité is one of the largest university hospitals in Europe. They cover all clinical care research and teaching, and the Charité Oncology and Hematology, a medical center, specializes in the treatment of leukemia, lymphoma, and solid tumors. And for them, innovative capacity and responsible governance for the benefit of patient and society are the central tenets behind all of the Charité research endeavors. A bit the same for the Ludwig Maximil University in Munich, our second German partner, and they are a very large hospital with 2,000 beds, highly advanced, and the 47 clinics, and it's in fact one of the largest hospitals in Europe. There is a special interest in molecular pathogenesis of leukemia and lymphoma, as well as in innovative treatment strategies. LMU played a leading role in the establishment of the standards for care for MCL patients. Now, these were, in a nutshell, our buyers. Now, the process itself, at least 4 proposals will be selected. 4 proposals will be selected for proving their solution is realistic. They will have to propose for that their methodology on analytical validity, clinical validity, their plan for clinical validity, their business plan, their IP strategy, IVD compliance. And in principle, from these 4 suppliers, we will select 3 who then can demonstrate the analytical validity of their proposed solution. And finally, from these 3, 2 will be selected to also demonstrate the clinical validity. So at the end of the process, 2 solutions should be retained as potential candidates for further development of their solution into a marketable product. So in the next slide, I will go a little bit more into detail with the project. We are now today just before, let's say, the opening of the tender. And as you already understood, the aim of this procurement is to trigger the development of new solutions and demonstrate the coverage of broad mutation profiling in liquid-based samples from, in principle, solid tumors, but the lymphoma are considered a solid tumor within the lymphoma. So the key players, on the one hand, the buyers or procurers, and the other hand, the solution developers. We, Sensano, are the lead procurers, and the buyers group I've just explained. Now, in phase 0, as indicated in the slide, the buyers have defined what their unmet need is, complete their requirements, and we are now preparing for the call for tender. The content of the tender is a consensus between all the buyers, and in addition to preparing that, we also prepare the business case for convincing our payer organizations to step into the oncNGS solutions or the opportunities that it offers. For the tender, we have described in very much detail the technical specifications, and we also have come up with some GDPR or some advice for GDPR ruling. We also discussed on the IVD implementation, and we also looked at the IP issues that may be linked to such a solution. There are 3 phases, and as I said, Rosanna Alessandrella will go in much more detail. The first phase is in fact the design assessment. This will take about 6 months. The phase 2 is covering the analytical validity. We will foresee about 1.5 years for that. And then in phase 3, there is the clinical validity, and there you have about 24 months to complete this phase. And at the end, we hope to retain 2 valuable solutions. The deployment of the NGS, well, what we hope to come to launching such a project is at the end to come to more common guidelines on implementing the oncNGS solution, or maybe broader seen on implementing NGS analysis. We should come with protocols of data sharing at hospital level. It may incentivize countries to launch cross-border purchase procedures. We should allow the development of interactive e-consulting facilities. For example, joint molecular tumor boards. And I think in the age of COVID, many people of you have already got familiar with such e-consulting. We could also start thinking on multicentric clinical trials and definitely having one solution for different hospitals. The same solution for different hospitals will facilitate setting up such trials. And then, as I already explained, we could come to some patient matching too. Now, as I said, we need to develop some business cases. Well, I just give you one case on how oncNGS could, let's say, have an impact on what is going on in a particular country. And I took Belgium as a test case, because due to the COVID crisis, many of the people that normally we have to contact and discuss with are very much occupied with all the aspects of the COVID crisis. So we will proceed on doing that in the coming years. So we will come back in more details on these things. So similar exercises will be done for the countries, for the other partnering countries. So in Belgium, the diagnostic testing in oncology is managed within the National Sickness Insurance, together with the oncology health professionals. For NGS in oncology, Belgium has installed a specific process wherein the scope of testing is agreed at the level of a newly formed ad hoc commission, which is called the Commission of Personalized Medicine, or the COMPERMED. They decide on which markers are to be included in an NGS that can be reimbursed by the NIHD. But of course, we have to pass also the financial restrictions. So there is some evaluation on the sustainability of this new testing. And NGS testing is to be accredited in Belgium under ISO 15189. It has to be for that an approval. It receives for that an approval by our national accreditation body, BELAC. And labs are obliged to participate in the national NGS EQA program. Labs can choose more or less till now what to use as testing. They're also free to develop their bioinformatics pipeline. The interpretation is on their responsibility and reporting, but we try to now, with the COMPERMED, streamline that process very much. In 2022, there will be a major impact on by the fact that the IVD regulation is coming into full operational action. And the COMPERMED, as I said, is trying to streamline these processes to make that we get more standardization and harmonization in NGS diagnostics in oncology. Now, it's clear that when you would implement such an oncNGS solution at the national level, that would be somewhat disruptive to the system, as the full benefit of such a solution would come from a general implementation of it at the national level. Cost reduction would probably demand the installation of a federal procurement instance for medical diagnostics, which is at the moment not existing, but we are discussing on that altogether at the COMPERMED and with the different governmental responsibles. The oncNGS market in Belgium, well, we have about 11 million inhabitants. There are about 70 new cases of cancer per year, and it's rising. And the reimbursement, as I already said, is managed by NIHD, or the Risif Inami in Dutch and French. We have an estimation today of about 12,000 tests for NGS in oncology per year. The reimbursement fee is set at €350. And with respect to liquid biopsies, we haven't really an agreed scope today, but roughly estimated, we probably would envisage between 5,000 to 10,000 tests a year beginning. So if we would extrapolate that to the 5 bio countries, which represent about 200 million Europeans, this would amount to about 100 to 200,000 tests a year. Now, if you look at the oncNGS market in total, if we look at, let's say, the US and the big 5 in Europe, there are various numbers that are being put forward, but the market in total for oncology is about 120 billion euro. Also in Japan and in China, this market is rising and is already expected to amount to more than 20 billion by 22. And if you look in that, of course, the major cost in the oncology is care and the medicines. But if you look at an estimation on the next generation sequencing market for oncology, it would come to about 250, 260, 300 million in 2020, but it's expected to grow exponentially to about 810 to 1 billion euro a year in the future. This is more or less what I wanted to say. Thank you.
Okay, so good evening, everyone. I'm Luca Mazzarella. I'm an MD PhD. I'm the head of the Translational Oncology Lab at the European Institute of Oncology in Milan and the coordinator of the genomics working group of the Alleanza contro il Cancro consortium of 26 cancer-oriented hospitals in Italy. My point today is going to be to illustrate the advantages and ongoing issues in the implementation of liquid biopsies. And these are briefly listed in the slide that I am showing here. And we will now briefly go through each of these points. So for sure, I will start by pointing out that liquid biopsies are increasingly there is mounting evidence on how this technology can really provide precious information for the identification of patients that are that maintain minimal residual disease after macroscopically radical surgery. And this has actually been demonstrated in a number of trials in various diseases. Here I'm listing trials showing the recurrence-free survival in major diseases like breast, lung cancer, and colorectal cancer, where you can clearly see that the almost 100% of the patients that show persistent minimal residual disease detected by liquid biopsy after surgery relapse within the follow-up time frame, whereas hardly anyone with a negative circulating tumor DNA will relapse over the months and years to come. So this is clearly likely to greatly overperform any predictive model that is currently in place for the prediction of the risk of relapse. In the metastatic setting, it is also proving of great advantage, and it is actually an established technology for decision making, especially in the context of lung cancer, where the liquid biopsy for the T790M mutation of EGFR is now an approved biomarker for switching to last generation tyrosine kinase inhibitors. But beyond these indications, also additional types of genetic alterations, like for instance non-single nucleotide variations like copy number alterations or splicing isoforms, etc., are being used to monitor the disease response to treatment in the metastatic setting, and they represent certainly an area of valuable improvement in the future. Besides these, if you want, technical advantages over established technologies, there is, of course, as we all know, one major factor that is key in the evaluation of liquid biopsies nowadays, and is the non-invasiveness of the technology. Clearly, this is a technology that has advantages for the patients, and this, beyond being obvious, is now also evidence-based, as this little but interesting study from the Princess Margaret shows. The vast majority of patients, of course, at this institution preferred the blood tests over tissue biopsy, as you see in this table, but they were even agreeing to trade some sensitivity in the test in order to have minimal invasiveness, and this is highlighted by the 6.2% difference that was deemed tolerated before switching to the liquid biopsy from the solid biopsy. But beyond these two major advantages, so the non-invasiveness and the superior sensitivity in some specific settings, there are a number of issues. And in my personal opinion, most of the issues they boil down to one specific point, the issue of size. How big should a liquid biopsy panel be? Of course, we can opt for a small panel that only looks at few alterations. This is quick, it's simple and inexpensive, but of course this has very limited applicability, and this is of course the case, for instance, of the EGFR T790M alteration. Of course, a large liquid biopsy panel would be much more versatile. It could be used for many more clinical indications, but at the moment these panels are slow, complex and expensive, and they can only be implemented in some specific settings. And these may also be suitable for different types of alterations that are non-canonical for liquid biopsy, like copy number variation, the additional issues on liquid biopsy, they boil down to this particular problem. But let's look at the other ones. Turnaround time. So turnaround time at the moment is a key determinant for for instance, the enrollment of patients in clinical trials, as you can see in the graph on the right that analyzed the reasons for failed enrollment in a molecular tumor board in the States, where one of the key issues for non-enrolling patients with a positive indication for targeted therapy was the time to decision that was heavily determined by the time it took for the genetic information to be obtained. And a recent small study presented at the World Cancer Lung Conference last year showed that there is clearly a halving of the time that it takes to obtain the results when you're implementing a liquid biopsy-based genetic testing in non-small cell lung cancer, as opposed to solid biopsy. And of course, this at the moment is an issue, because if you want to carry out an analysis with a large panel, you have to send your sample off to a centralized laboratory, with all the turnaround time issue that this may pose. But of course, as we will see in the last slide, a different model for the implementation of liquid biopsy may contribute to reducing this key point. And in this slide, we also see data from a recent trial in gastric cancer, where you clearly see that two trials with very similar indications, in which in one case in the GA screen, the enrollment was based on tissue biopsy, whereas in the Godzilla trial, enrollment was based on ctDNA. The time to enrollment was reduced from 35 days to 10 days. And as you can see in the graph on the right, you clearly see that the red line, which is the enrollment from the moment Godzilla was launched, skyrocketed the moment the ctDNA based trial was started. Cost then is the other issue, and this again has to do with the size of the panel that we want to implement. This is a recent analysis carried out by an Italian group in which they explored different models for managing patients in the first and second line of non-small cell lung cancer, in which they explored the health economics aspects of a completely tissue-based strategy, which is the more orthodox strategy being implemented nowadays, a mixed strategy that is also what has been happening in the last couple of years, where you use liquid biopsy in the first and second line, and a potential strategy in which you try to base all possible decisions firstly on liquid biopsy and only if necessary on tissue biopsy. And to cut the story short, the message was that the combined strategy and the potential strategy, so both strategies based on liquid biopsy, were significantly more cost effective. But, and this is a major issue in the point, this was only if the cost of the liquid biopsy was kept at the current standards for a single gene cost, 250 euros more or less. Of course, with liquid biopsy panels where the size and the cost is considerably higher, this is certainly not the case. And this directly poses an issue of equality of access. There are very few studies on this point. There is one recent study that was conducted in Colombia that, of course, may not be considered a representative nation for healthcare systems like those in Europe, but at the same time, the considerations are in Europe are very similar to the ones posed in this particular paper, in which in the end the use of HER2 monitoring using liquid biopsy in the management of HER2 positive breast cancer was considered basically not effective for the Colombian healthcare system and not applicable. So therefore, if we want to have a large-scale implementation of liquid biopsy, the reduction in cost is an imperative. Otherwise, this will be limited to very specific context of very specific patient population with a very low equality of access. Another issue that is common with solid tumor testing is the issue of the standardization of the reports. So the amount of information that is collected in these studies is very high, and this paper from the American College of Pathologists a few years ago compared the agreement on the types of information that would be reported in a typical report from 44 molecular pathology labs in the States. And as you can see, the level of heterogeneity for different categories of information was very, very high. But lastly, we really think that a more widespread implementation of liquid biopsy, especially in the metastatic setting, may really be key to increasing the access of patients to clinical trials with targeted therapy. There's a number of designs that can be foreseen where you use liquid biopsy. You can use it to pre-screen, you can use to screen, you can use to monitor the disease, etc., etc. But importantly, there already is evidence that when you provide clinical recommendations based solely on liquid biopsy, either because there is no information coming from the solid biopsy, or because there is even disagreement between the solid biopsy and the liquid biopsy, maybe due to clonal heterogeneity of the tumor, then in a vast majority of the cases, you do provide information that is valuable from the clinical standpoint, because as this very nice study on JAMA Oncology showed, you can induce responses, meaningful responses, when you're basing your treatment solely on the liquid biopsy. And finally, a major issue is the model of implementation of liquid biopsy. As we briefly said at the beginning, at the moment, you can easily implement a small-sized liquid biopsy panels locally, so every lab can run a droplet digital PCR on EGFR T790M, for instance, but then larger panels that are based on NGS are at the moment are mostly implemented centrally by large companies that, of course, aim to rationalize the resources and to obtain, to increase the database of background information to provide more accurate results. However, this is a model that, of course, is going to be optimal in the initial phases of the worldwide implementation of liquid biopsy. In the present context where demand is growing and it's probably not going to be met in the best possible way by an entirely centralized model, this model should probably change. And even then, this model poses major issues on privacy and accountability, because there is, of course, a data transfer, especially in an ecosystem like the European one. With the advent of the GDPR, this may be a major issue, a major obstacle to the implementation of large-scale liquid biopsy. This, of course, poses logistics problems. You have to organize the shipment of the samples, and the samples may degrade, then may be lost, there may be a number of problems, and of course this is going to increase turnaround time. Of course, for low volume centers, this is still the centralized model is still a preferred option, but for large volumes, for large volume centers that can sustain a certain volume of analysis, then turnaround time is likely to be reduced if the analysis is carried out internally. Of course, when we are talking about decentralization, doesn't necessarily mean that every single hospital will need to carry out their own testing. It may also mean the transition towards a hybrid, if you want, system where a set of hubs is defined as the centers where the assay can be run for a number of spoke hospitals in the network. So in conclusion, what we think is that the liquid biopsy-based disease assessment in principle is superior to tissue-based biopsy for a number of reasons, and therefore it's highly desirable. It's better tolerated by patients, it's more accurate in detecting MRD, it's very highly sensitive in monitoring metastatic disease, may be more sensitive in identifying resistance, and it may be also very useful in allocating patients, potentially even more effective than current methods. However, there remain major issues regarding the panel size and the range of alterations that can be detected, CNV and translocations. Probably Pierre is going to talk a little bit about that after me. And this also poses a major issue on cost and sustainability for the value-based healthcare systems that we have in most European nations. And that also means that the size of the target population and the quality of access is compromised. And we strongly favor the implementation of a local model over the implementation of a centralized model that is the dominant one for large panels at the moment. And with this, I'm done. Thanks a lot.
Hello, my name is Pierre Sujaubert. I am a molecular biologist working in the hematology lab from the Hospice Civil de Lyon in France. And today I will present to you the technical challenges of the oncNGS proposal, which can be summarized as the following goal: bringing together what has been separated. As you know, cancer genome is not so easy to study, especially because of spatial heterogeneity, meaning that depending on the site that you study, you will not find the same molecular abnormalities. Moreover, cancer genome is not the same during time because of Darwinian evolution of cancers during time. And we really need to monitor treatment response to detect residual disease and to detect as early as possible relapses in order to propose preemptive treatment when available before the patients develop clinical signs and symptoms due to relapse. So basically, if you compare the advantages of solid and liquid biopsies, solid biopsies obviously will remain very important because they are the only one to get histological analysis of tissue, and this is the gold standard of diagnosis of cancers. However, for all the other features, liquid biopsies seems to be very interesting to resolve spatial heterogeneity, to identify targets. The material is very easily available due to simple blood sampling. You can repeat this measure as much as you want. And this is very easy to standardize, probably more than for histological analysis. However, there are a lot of technical challenges before liquid biopsy can become the standard of treatment of cancer. First, we need to optimize the pre-analytics, meaning that we need to obtain very good quality ctDNA. It means that we have to make some choice, such as what to sample, blood sample or other fluids, at what time of the treatment or at what time of the day we have to sample the patients. And there are specific pre-analytical constraints on ctDNA analysis. We need absolutely to avoid white blood cell lysis in order to avoid contamination of cell-free DNA by this DNA. So you need to choose the good sampling tube, to choose the good transportation system. You need to separate as fast as possible the plasma from the blood cells. ctDNA extraction, quantification, and conservation are also specific points that need to be optimized in order to optimize the analysis of ctDNA. The goal of the oncNGS PCP is to develop the best solution for the analytic part of liquid biopsies. It means that all these features have to be optimized. We want a panel which is as much informative as possible, which means that for all patients tested, we want to find a marker to help for the diagnosis of cancer. We need a sensitive solution able to detect variants with very low allelic frequency. The solution has to be reproducible, robust. We need as limited wet lab handling as possible. The bioinformatics should be standardized as well as the clinical report. In order to be clinically useful, the time to result should be less than 10 days because the clinicians will expect the result to decipher what treatment they will give to their patients. And in order to be clinically useful for all the citizens of the European community, the price has to be reasonable in order to make it accessible for all citizens. Finally, flexibility is important because the solution should fit for one sample and also for many samples, and it should also be flexible to adapt the panel to scientific evolutions. So our principal research and development needs are the following. We need a state-of-the-art panel design with maximal informativity with all the clinically relevant targets regarding the diagnostic, the predictive and prognostic biomarkers, and the biomarkers of theranostic implication. The price, once again, has to be in line with standard of care applications. The wet lab should be automatizable with internal quality control and a limited time on hand. And bioinformatics should be standardized, open access, with data interoperability, databases reconstruction, and all, of course, should be compliant with the GDPR regulation of the European Community. There is a huge market opportunity regarding ctDNA analysis because it will become the standard of care for the analysis of the molecular landscape of advanced cancers and lymphomas. We want to develop personalized medicine, meaning that for each cancer we will have one molecular analysis in order to decipher what treatment we should give to the patient. And finally, ctDNA will be very important for the longitudinal monitoring of cancer patients in order to follow the treatment response and the relapses as early as possible. So I thank you very much for your attention, and will be available for the question and answer session if you have some point to discuss with me. Thank you very much.
So, hi to everybody. Here is Rossana Sandrello from AQUAS, the Agency for Quality and Assessment, Health Assessment of Catalonia from Spain. And thanks you for joining this Open Market Consultation. We are really excited to go through this event with the oncNGS consortium. It's as in any other PCP process. The Open Market Consultation is a key event to gather to introduce the oncNGS objectives, the PCP objectives, but also to somehow ensure that the market is understanding us, understand where are we going, what we want to achieve with this pre-commercial procurement, and consequently, why not to engage with the buyers for a fruitful collaboration later on. So let's start. Okay, the very first question we would like to address is why an open market consultation? As I said before, for the buyers group of oncNGS, the market consultation is an opportunity to double check if the minimum requirements of the innovative solution they are pushing, they are willing to achieve to address their unmet needs is aligned with the market maturity, and the market is ready to satisfy them. Also to double check if everything is clear from a regulatory standardization perspective. The assumption that the buyers are doing to make this happen, sustainable in the time, and that the implementation deployment one day of oncNGS solution makes sense, is feasible, is what we call business case. And we need to make sure that the market understand what are the long-term vision of the business case, and if these assumptions that we are doing for the business case are achievable. And at the end, also the feasibility that this procurement makes sense from a market perspective. The market. No solution can be provided to the unmet needs of oncNGS if the market is not engaged in this procurement. So this alignment between the buyers and the market is key. At the end, the methodology of the open market consultation will consist in 3 steps process as announced in the prior information notice, the open events that are taking place today on May 11th and May 12th, the questionnaires that will be launched just after these 2 events, and will need to be answered in the almost 1 week time. And then the online meetings with each of the respondents to the questionnaire. We have a deadline also for the meetings, but also we need to make sure that we will give an answer, we will give a slot to each of the respondents. So at least we know that we will start on May 21st. The procedure, as you can see already, is a long procedure. This length of the procedure is not due to the fact that that we are that we from the very beginning we are buying something that is off the shelf available. Is due mainly to the fact that we are the buyers group is now committed, is going to invest in research and development services. Consequently, it will be a 3-phase process, a competitive process. At each phase, there will be a competition to move on to the next phase. And this implies clearly that a very detailed exercise to regulate all these steps in a way that all of us will feel comfortable, the buyers from one side and the economic operators will join the PCP from the other side. So everything started with the prior information notice publication. Then there has been there will be the market consultation, that is the phase we are now. And after the market consultation, we'll have the contract notice at the OGU, where we will launch the contract. We will issue the invitation to tender, request for tender. And in the request for tender, mainly there will be a set of documents. Part will describe the administrative procedure and all the regulations somehow that regulate the procedure. And on the other side, there will be the challenge brief, the technical specifications, and that will describe the unmet needs, the common unmet needs identified by this big buyer group, the challenge, how they feel that the challenge can be addressed, thanks to all the work they are doing still are completing in this month, not only internal interviews at each buyer side, but also literature review, interviews with experts, and so the concept validation they did across all this month. And lastly, also the business case. That is somehow the business opportunity for the economic operators that are going to engage. Because if the economic operators are unclear why the buyers are making such investment in oncNGS, there also will be clear what will be the business in the future. So as I said, then there is a 3-phase process. The tenders will be submitted from the very beginning, knowing how it will be, what we are expecting in phase 1, in phase 2, and phase 3. How we will assess the offers, how we will what deliverables are expected in each of the phases, what milestones, how the contracts will be monitored. So everything will be clear from the very beginning when the bidders will submit an offer to the tender. Once the evaluation of the offers is finalized, the 4 most advantage offers, so that they get the highest awarded scoring, will be awarded with a phase 1 contract, and we will go through the solution design that will last 6 months. And per bidder, per economic per awarded contract, this contract volume is valued approx 180,000 euro, VAT excluded. Then we have phase 2 that will take 12 months and will consist in the prototyping and analytical testing. And per bidder, the contract volume is valued 700,000, approximately 700,000 euros, VAT excluded. And at the end, we will have the clinical validation of a limited set of pre-commercial solutions. This will take 15 months, and the contract volume per contractor, per bidder, is valued around 2 million euros, VAT excluded. To go through from phase 1 to phase 2 to phase 3, the contracts that contractually will successful and satisfactorily complete their phase will be invited to submit a bid for the next phase, for the following phase. So there will be like an invitation, a mini tender called invitation to tender to next phase, to phase 2 or to phase 3. And again, the best offers will be awarded with the next phase contract. Consequently, along the process, the contractist, the awardees that will be awarded with phase 1 contract will be requested to sign not only phase 1 contract, but also a framework agreement. And this framework agreement will regulate how we will work across the 3 phases. So when we go to phase 2, again, we will be allowed to invite only the economic operators that join phase 1 and satisfactorily and successfully completed phase 1. No new operator will join phase 2 or phase 3. There will be the ones that entered in phase 1, okay? Another thing is that it's important to highlight that right now you are here, no? You are acting as participant to the market consultation. This means that the economic operators that are joining this phase are just here to share with us, with the buyers, their maturity level from technological perspective, from service perspective, their understanding on the business case, but they are not, it's not a preselection, no, to join this PCP, and they don't have any advantage, specific advantage to then be selected for the PCP. The bidders that will submit an offer to the PCP, they will be evaluated against the awarding criteria that will be defined in the request for tender and accordingly to these documents, and just only the ones that will get the will meet the minimum requirements, and only the one and will be ranked, the 4 most ranked economic operators, bidders will be then awarded with the phase 1 contract. So no pre-qualification, no advantage for joining the market consultation. Sorry, and just to add also, that's important, that every time that we have a bidder that then is selected to pass to the next phase, will become a contractor. But then when we end the phase 1 contracts, this economic operator that will be invited to submit an offer to phase 2 will be again a bidder. And if it is selected, will be a contractor. And if it is invited again, it will be a bidder, and then again will be a contractor. This is important also from regulation perspective, but depending which phase we are. If we are in the phase of the invitation request for tender, or an invitation to join this the following phase, or if we are in the contract phase. All this procedure is regulated in agreement with the Treaty of Function of European Union and the Public Procurement Principle. So we need to make sure in all the moments that these procedures started, remember, no? We said with the prime informator's notice, it's going to be transparent. We will treat equally and not discriminate any of the economic operator. We will recognize all these reports and certificate issues by different authorities in the different countries across the EU, EEA states. It will be an open competition, no restriction, no pre-qualification. We will be proportional, so the requirements and conditions of the procurement will be reasonable in proportion of the object of the procurement, and also, and that's very important, it will be managed soundly, financially sound. This means that all the contracts will be designed, awarded, and monitored in line with good principle of economic efficiency, effectiveness from public reason perspective. Say this, we can already tell you that that's very important, that we are now in a phase that all the audience here, but again, since we are now in a phase of an open market consultation, we can receive questions. Any of us can receive questions, but still, we thank you. We really thank you if we can use just one channel. That could be the Contact Us page in the oncNGS webpage. The Contact Us form is the preferred way to receive questions from you. And in this way, also we can guarantee that we can, all the questions and answers will be collected in one document called Frequent Asked Questions, and will be made public in our webpage. Don't, in any case, if we get inquiries different from the channel, again, all the questions and answers will be published in this document. This is important to ensure that we meet the public procurement principles that we were mentioning in the previous slide. Also very important that this pre-commercial procurement is funded by Horizon 2020, and so we need to ensure that this procedure will comply with Horizon 2020 rules. Some rules that now we're going through are key to understand how the pre-commercial procurement procedure will be designed. And so we want to make sure that the audience and the economic operators that are joining now the market consultation, or will join later on as a bidder, potential bidder, understand that these rules are there and will be followed by oncNGS procedure. We are going to buy R&D services. That is our object of the contract. It will be not supply, and it will not be services of another kind. It will be R&D. This means that more than 50% of the object of the contract will be devoted to research and development. And if it is not devoted to research and development, all the rest of this presentation does not apply any longer. So we need to make sure that we always are under this criteria, condition, that we are procuring R&D. No, we are not procuring innovative services. We are not procuring off-the-shelf solution. We are procuring R&D. And R&D needs to be demonstrated because it does not exist, and we are creating for this project. Risk-benefit sharing under market condition. That's very important for the economic operator that will join this procurement procedure, because this means that the buyers and the contractors are sharing risk and benefit. R&D, we already know that there is a risk coherent, no? Already beside the concept of R&D. And at the same time, what we are saying is that while the procurers share with the suppliers at the market price the benefits, are also these the IPR resulting from the R&D are shared. And now we are going to tell you how. First, before telling you how, let's also skip to the other point. That is competitive development in phases. We already told you, no, there will be a process in 3 phases. And so we are, and this will allow the buyers to compare, identify the best value for money solutions across the overall solution. And consequently, we have these 3 phases: solution design, prototyping, and original development and validation of the first prototypes. This means that we are clearly separating the R&D procurement, the R&D of services, from the deployment of commercial volumes of end products. This will come later, will be a PPI. Right now, we are just buying procurement of R&D services. The word just maybe is not just, because this is the really most critical part. But there is no now procurement of commercial volumes of end products, of end services. How we are sharing risk and benefits? Because the procurers do not reserve R&D results exclusively for their own use. I don't know if you are familiar with public procurement. When usually in public procurement, the procurers reserve the results at their own use. In the case of a PCP and in compliance with H2020 rules, the procurers do not reserve R&D results exclusively for their own use. And the R&D providers or the economic operators that will generate the IPRs then will own the attached IPR generated through this PCP. So what happens to the procurer? The procurers must enjoy royalty-free access, right to use the R&D results for their own use. Must also enjoy the right to grant or to require participating economic operators to grant no exclusive license to third party under fair and reasonable market condition without any right to sublicense. Okay, so we are talking about that at the end, the economic operator will own the attached APRs, and the procurers will enjoy the royalty-free access right to use the results for their own use. And then also the possibility to grant no exclusive license to third parties. There is also a callback provision. That callback provision is key. It's key for the procurers, because the procurers are making this investment. This investment means an effort from their side. So they don't want to have the fruit of the research and development then abandoned. Then no one is exploiting this R&D. Because if they are making this effort, it's because they want this R&D at the end used by their healthcare providers and used across Europe. So if at the end, if we, after a number of years, will be defined in the call for tender, the economic operator, the R&D provider, fails to commercially exploit the IPRs resulting by the PCP, or are using this IPR to the detriment of the public interest, the procurers have the right to call back these IPRs and attach this IPR to someone else, because at the end, the economic operator that generated these IPRs did not exploit the IPR. Clearly, this will be done in a given number of years, and the exploitation doesn't mean already to commercially sell it, but means, for instance, to maintain the APR, to maintain the licenses, to ensure the updates and backup of the technology. All of this is when clearly we see the R&D provider is aiming to commercially exploit this APR. Then again, when we are talking about sharing risk and benefit, we want to highlight another thing, that if the PCP procurers are will procure these R&D services at the market price, given the fact that they will not retain the ownership of the IPR as in any other public procurement procedure, they will in return they will have a financial compensation, and this financial compensation is somehow what compensate the procurer for not retaining the IPR. And the other side, the economic operator, thanks to this financial compensation that is given to the procurer, is retaining the attached IPR ownership. Okay, so and then in any case, as already we said, procurers must receive rights to use R&D results for internal use and licensing rights subject to certain conditions that will be defined in the public procurement. All of this bring to us a very important point that I also would like to bring here to all of you, so you understand that since we are procuring R&D, since we are sharing risk and benefit, since the IPR are at the end owned by by who generate the IPR, this PCP is exempted from the EU public procurement directives because the procurers do not retain all benefits of the R&D. Is exempted from the WTO government procurement agreement because we are procuring just R&D, and does not constitute state aid because it's a process, it is open, transparent, competitive, with risk and benefit sharing at the market price. So at the end, the bidders can be any type of natural legal persons. We will then clearly specify in the tender documents some selection criteria of the knowledge and experience related to the object of the contract. It will be possible to submit bids either individually or in association or consortium with other bidders. And clearly, no one will be allowed to submit one or more bid. The subcontracting will be possible, but limited to percentage. Also, this will be specified in the call for tender. The platform performance, there will be more than 50% of the R&D performance will be performed in European member states or Horizon 2020, associated countries, and the language at least will be English. So what happens then next? Next, we will have, once we have completed the PCP and we are successful and we are happy and the buyers are enthusiastic of the results of phase 3. I'm now somehow joking, but this should be what we want, no? That this investment bring valuable solutions to one day be deployed at the buyer side. Means if this happens in reality, the buyers so are satisfied with the solution and the feasibility to deploy them, to deploy the solution at their site, at their premises, then there will be a post-oncNGS procedure. And this is the one when finally the PPI, the oncNGS commercial services will be contracted. no phases. It will be a normal public procurement because it will be under all the exemption they were applying before. Now there will be no exemption. And the most important aspect is that part of the technical specification of the challenge of the PPI clearly refers to the original oncNGS challenge technical specification, because it's exactly PPI fruit of the original PCP. And so it will be an open procedure, and where it will be possible then to finally deploy the fruits of the PCP in the real clinical practice. Okay, so this is my slide, last slide. Thank you very much for listening to me, and don't hesitate to make any questions, to submit any questions through the contact form of in oncNGS page.